Hello, my name is Ron Kohanski. I am the director of the Division of Aging Biology at the National Institute on Aging. Thank you to the organizers of the AGE meeting for giving me the opportunity to speak. I'll talk a little bit about funding at the National Institute on Aging and then make a brief introduction to the Division of Aging Biology. So, getting funded at the National Institute on Aging is somewhat similar to getting funded at any of the NIH institutes. There's a site on the NIH website, grants.nih.gov. Link on these slides are active. And this link will take you to the NIH grants and funding site. There are descriptions about the grants process, getting started, how to apply. Policy and compliance is something that you do need to pay attention to. There are policy changes on submission deadlines, submission procedures, number of pages that are allowed for each grant, information like that. And there's information for you, the investigator, and for your administrators on the submission of applications. At the National Institute on Aging, there's a landing page. You can always just search National Institute on Aging, and this will be the website that you'll get. There are lots of different aspects here to the website. The one that you'd be most interested if you're looking for research and funding would be the research and funding drop-down menu. And this link, Research Division and Contacts, will take you to a list of the 4 scientific divisions at the National Institute on Aging. You can also see some special announcements that we have on grants and funding, which I'll go over a bit later. There's also training and career development grants. I'll mention those as well. Information about clinical trials that the NIA supports and research resources. You may be familiar with the NIA-supported rodent colonies, where you can get aged C57 black 6 mice and aged Fisher 344 rats. And there are also access to non-human primates and biorepositories from various rodents and non-human primates. So you're going to write a grant, which technically is called an application. And in writing a grant, you've heard a lot of advice. I will just add a little bit to it. When you write a grant, you need to state what you mean. It's very important to state your hypothesis clearly. The majority of research supported by the National Institutes of Health is hypothesis testing research, which means technically you might be mistaken in your hypothesis. The data might not support it, in which case you would need to have alternatives if your hypothesis is not supported. And those have to be real alternatives. Statements such as, we've been doing these experiments for a long time and we expect them to work, it might not be your best option. You have to give actual hypotheses, real tests of those, rigorous tests, and suggest alternatives either in the experimental approach or in the alternatives to the hypothesis themselves. And you also might want to suggest next steps. So when you write a grant, you don't talk about what you're not going to do. You talk about what you will do. But if your research is successful and gives you information about the system you're studying, you might suggest what you might do as a follow-up to the research you proposed in the grant. Don't be overly ambitious. Most R01 grants are 3 specific aims. R21 grants or R03 grants are usually 2 aims each. If you need more, don't get carried away. And if you need fewer, be cautious. You need to figure out how long it takes to do the research and or estimate, and then make a proposal that's in keeping with that. So you write simply. You need to come to the point. Think of writing like Ernest Hemingway, short declarative sentences. You're not writing like Jose Saramago. They were both Nobel laureates, but Saramago can write a sentence that goes on for 10 pages. You don't want to do that. And if you have a lot of abbreviations in your grant, you might want to have a table of those abbreviations accessible. You might be using non-standard abbreviations, and the reviewers would help that. Basically, you're writing the grant to help the reviewer evaluate your proposal. So you want them to understand what you're writing. They want you to agree that it's important. You want them to agree that it's important. And you want them to appreciate the context in which you're doing the research. So there are a bunch of puzzles at the NIH. We are a bureaucracy, and we use a lot of abbreviations, and we use a lot of arcane language, if you will. So you'll get these, you'll submit your grant. Your Grants and Contracts Management Office will submit it on your behalf. Technically, it's the institution you're working at that would get the award because they have the resources to accept the money and disperse the money. They also own the loading dock where you might get your equipment supplied. And they're responsible for your salary as well. So the grant actually goes to the institution, but you think of it as yours. Good idea. So in these, you'll get a grant number, and it has a lot of information in it. The first is the type, and there's the mechanism, the institute at the NIH that's providing the overview of your grant. They accept it for review. There's a tracking number, year of the grant, and some qualifiers. So the types, it's useful to know this. There are three major types of applications that you'll be making: new, renewal, and revision. New application is the first time you make it. If you get the award, toward the end of that grant, you submit to get a continuation of that. That's called a renewal. If in the course of the research you decide that you need to expand the scope of the grant, you would make what's called a competing revision. Competing means it will go for peer review. Revision is you're changing some aspect of the grant or adding to it. Under revisions, there's also administrative supplements, which are not peer reviewed, but are reviewed by program officers. These others are less often encountered. Type 4 is an extension of a grant. They occur in rare circumstances. The 5 indicates it's a non-competing grant, which means you've been given the award. You're not competing for the funds. We've already agreed that you'll be getting them in the first and subsequent years. And then these others, 6, 7, 8, 9 are for change of institution. These are always single digits. Next thing is the type of grant. Briefly, these are the basic research grant, which is a 5-year, up to 5 years in general. And 23 NIH institutes and centers participate in these grants. The R03 is a 2-year small grant that's limited in dollars. And not all NIH institutes support this mechanism, as it's called. Only 11 NIH institutes and centers support it. So if you wanted to submit, for example, to National Heart, Lung, and Blood Institute, they do not support what's called an investigator-initiated grant that's an R03 or an R21. They'll only accept those grants if they issue a special request on a special topic. So you have to check. If you write up a grant and you find out you're targeting it for an institute and you find out they don't accept it, you would have to redirect the focus of the grant to another institute or find a different mechanism that your preferred institute supports. So the R21 is a 3-year exploratory grant. Only 15 NIH institutes and centers support that. Not everybody supports a program project also, but there are special announcements, funding opportunity announcements for these. These are typically 5-year program projects involving 3 or sometimes more investigators, 3 projects, each of which could be independent or submitted as an R1. But there's a synergy among the projects so that the outcome from the whole program would be greater than the sum of the parts if considered individually. The U01, the U as the first letter indicates a cooperative agreement. A cooperative agreement means that there would be engagement by some federal employee in the workings of the grant, either a science officer who's technically going to be a program officer, but would be engaged more in the design and execution of the research, attending lab meetings, offering advice, or you might have a collaboration with one of the NIH intramural investigators, or another NIH or federal entity. U19 is like a program project in some respects, except that the individual components of it would not function well if supported separately. And not all institutes support the U19. Then there are the K awards, which are mentored research awards. Then again, you have to check to see which institutes support these. And 9 support the clinical oriented K01. 6 support those that require human subjects to be involved in the research. So you would have humans participating as subjects in the study. And the basic research, which could also be somewhat clinically oriented, but it's not a trial, wouldn't allow trials. 17 NIH institutes and centers support those. Almost everybody supports the K99R00, which is a transition from 1 or 2 years of postdoctoral work to 3 years of independent research. Generally speaking, you're making a transition from the institution where your postdoctoral work is done to another institution where you have a tenure track or equivalent faculty appointment. And these also break down into no trial, trials allowed, or humans required, and different NIH institutes do or do not support those mechanisms. And of course, there's the famous 2-letter code for which institute. This is AG, NIA. There are also NIH roadmap programs, which are special programs. And you search NIH roadmap, and that will take you to the roadmap site where they'll tell you what kinds of programs those are. And then there are Office of the NIH Director, that's an OD. There are special programs there as well, transformative R1s, things of that nature. Over here, this will indicate the year of the grant. So if it's your first time through, it'll be a 01. The first non-competing year is 02, etc. If you get the renewal, that's going to be either year 6 on a 5-year award or year 7. If you have a no-cost extension at the end of your award, meaning you still have money left, but you're going to continue work on it, you can have that no-cost extension for the first post-award year automatically. The grant number then shifts to 06. If you are submitting a renewal and you get it funded, then it would probably be year 7 in that situation. But there are qualifiers here, which are for resubmissions. The NIH allows an initial application, technically in a 0, but that's not shown up as a qualifier. It would just be 01. 06. So if a resubmission would be a 1, there is no second resubmission, although after this one, if this one fails to make it to payment by an NIH institute, you would have to submit as a new grant. So it might be the same topic, same aims, written in a way that would appeal more to reviewers, but it would get a new grant number. And then it would be a 01 and not an A1. And then, of course, there are supplements. So if you've done one of those revisions, which would be a 3, R1, et cetera, 0, let's say 2, S1. So you could have submissions that run for the duration of, supplements that run for the duration of the grant, and the years would track with that accordingly. So that's the arcana of this, but it's important to understand one thing about it. Well, if you get funded from the NIH, you want to retain your funding, and it's very important that you do the following. Get people to work in your lab. It's not necessarily the easiest transition after you've been a postdoctoral fellow to attract people, but when you look for your faculty position, you want to know a few things. First of all, with whom will you talk about your research? You want to have people who are willing to talk to you about what you do. They don't have to be in the same field. But they do have to listen and give some reaction and some feedback to your interesting projects. So you want people to work in your lab. You also want to recruit for diversity in the workforce. There are opportunities to support people in your lab through the same sorts of things you might be supported under as a student or as a postdoctoral fellow, institutional training grants, individual training grants, individual mentored awards, and awards to support diversity in the workforce. So you need to contact your program officer if you have any questions about what those are and how to apply for them. It's important to continue to attend meetings, or if you haven't done much of that, to get out there and make yourself known. Peer review means that people who are in your field, presumably, will be reviewing your grants, and it always helps that they know who you are, that they get a feel for who you are from conversations with you, from presentations you make at meetings, from questions you ask at meetings, and just the socializing with other scientists. It helps. A reviewer cannot bring into the review information outside, but they know who you are and it has an impact. And you write more grants. You get your one grant, you get some work done on it. You try to write another grant off cycles, so you'll have grants at years 1 and 3 of your first faculty appointment, or years 2 and 4, which helps to keep things going. It's also useful to try to join with other investigators on their grants as either key personnel or as a co-investigator. It helps, of course, it depends on the culture of the institution you're working at as to how much they value your changing the direction of the field all on your own, or how much they value your collaborative work and your contributions to the scientific enterprise in general. You must publish. You must publish, and you need to get solid work out. It's not necessary that everything be published in the premier journals, Cell Science and Nature, but at most places, they would like you to be publishing in journals that do not have the title of your field. So you want to have some publications in general interest journals. And when you publish, you need to cite your grant support. And the part of the grant number that you cite is this: the mechanism, the institute, and the tracking number. This information, the type, the year, and the qualifier are not relevant, really. It's this part. It's important because we track your productivity through citations of the grant number. Now, there are more sophisticated ways for us to track it. You have to submit progress reports every year in which you identify your publications. And your publications have to conform to open access rules. So we do have that information, but this is another way in which we track your productivity. It's a metric. It's not necessarily the only one. It's not necessarily the best one, but it is important that you acknowledge that we've supported you. It's also on that last slide in your talk. You thank the NIH for their generous support. It's always good to be polite. So what grant should you write? Well, the way it works in the federal government is if you want money from us, you have to cite a funding opportunity announcement that's related to that. So, or there is such a thing as a notice of scientific interest, which will usually link to a specific funding opportunity announcement. So there's the investigator-initiated application. That's most of what the NIH sees. And those are in response to a parent announcement. And on this slide at the bottom, there's a link to the parent announcements, which has important information. Links to the forms, it links to the rules for submitting a different type of grant. And it also links to the deadlines. You have no choice but to conform to the deadlines. There are some few exceptions, but by and large, just conform to the deadline. And that means that you'd like to submit 1 week ahead. You want to make sure that the application gets into your grants management office in plenty of time for them to submit. And if there are any glitches or anything that needs correction, you would leave yourself at least a few days to clean up anything that needs to be done. You have to conform to all the detailed rules of formatting an application. So the kinds of funding opportunity announcements called FOAs. There's a request for applications. We often refer to them as RFAs. You'll pick up the language as you go along. Notices of scientific interest will indicate that we're not putting any money down on that, but unless we're joining an RFA, in which case we are setting aside money to support grants. But a notice of scientific interest can just refer to a topic that we'd like you to work on. We think it's important, but we don't happen to have money at the moment to support it. So we might link to a parent announcement, or it might link to a program announcement with review, which means that there's a special review group convened for applications that come in. So an RFA will typically have a single due date. Program announcements with review will often have multiple due dates. And then there's a program announcement with a set aside, and that differs primarily in that it will have multiple due dates for grants, in contrast to the RFAs, which by and large have single application deadlines. They may have more, but that's rare. So as stated, the RFA will have a single due date. The investigator-initiated applications are of three types, of course: new, renewal, and revision. RFAs might accept, for example, only new grants, or they might accept only revisions. Not that common, but sometimes the case. The NOCI will refer to another funding opportunity announcement, and then these have multiple due dates with a special emphasis panel, referred to as a SEP. And there may be agreements with the Center for Scientific Review, an independent body at the NIH, that they will handle the review on behalf of an institute. On what type of grant should you write? Depends on where you are in your career. If you're a PhD candidate or a postdoctoral fellow, or you are looking to be supported as following under one of the definitions of diversity in the workforce, might be interested in an F fellowship. If you're doing mentored research, you'd probably be a junior faculty member looking for a K award, or a transition from postdoc to junior faculty, which is the K99. There are also there's an F99, which is a transition from the PhD to the postdoctoral fellowship. And these can have very special rules about the length of time between your PhD or MD or other advanced degree. How much time can you spend in that fellowship and still qualify for the K99, our 0-0 mechanism? There are exceptions, of course, but you'd have to contact the program officer or scientific review officer to learn what those are. The language is clear to us, but it might not be clear to you. So we can help you with that. The research program grant will support your lab. The program project will be joining forces with other investigators, and the U mechanisms are cooperative agreements. There is no parent announcement for these. These are special FOAs that will request them. Or if you are going to work with, let's say, an intramural research program investigator, you would submit the application as an R type, and then if awarded, it would be converted to a U, if you're doing it as a investigator-initiated application. And as mentioned before, the U mechanisms have greater engagement and oversight from NIH program officers. And Bida Nakhai has made a presentation with more detail on grant writing and other aspects of it. But one thing I want to mention here is there are cycles for grant submission, considering the investigator-initiated applications primarily, but all applications fall into these cycles. The due dates vary, but they are strict in each of the three cycles. The application is accepted for review, and so the scientific review will typically follow anywhere from 1 to 7 months after the receipt. The advisory council is the final level of review before your a decision is made about funding your application. So these can be, for the first cycle, it's the councils go somewhere between August and October. Cycle 2, it'll be the January council. And then the earliest project start date will be after that. So if you have a May submission, you might not see funding for 11 months, or as long as, well, let's say 11 months. So at many institutes, the funding decisions are not made at the time of advisory council. Advisory council will have oversight over the review process, that they may concur with the review, or they may choose to disagree with some aspect of review on one or more grants or applications. But the funding decisions are actually made by NIH Institute directors with advice from program officers. So for us, for January council, we will have a funding meeting in March. We will finalize funding meeting in February and finalize funding decisions in March, and hope to get the awards made in April. So you have to keep it in mind. When do you contact your program officer after review? So you contact us before the application to discuss various aspects of it. Is your application suitable for our institute? If you really want our institute, how might you adjust it to be more suitable? And then after the review, between the submission and the review, you're in the hands of the scientific review officer. And then after the summary statement is released, you talk to your program officer, who helps you read the tea leaves of the review, the summary statement, and they help guide you in how to structure a response to a critique. Or in some cases, we at NIA ask for a response to the review, even if we're planning to fund it. So we might want you to pick up on a couple of points that they've raised as concerns. They're probably minor concerns if we're choosing to fund it, just so that we're reassured that you've taken them into consideration and have ways of dealing with them. So the program officer will answer questions about the science and give you advice on study sections. We might occasionally refer you to the scientific review officer to contact them. Do they feel that they have the expertise to review your application? And then the summary statement. You also fill out progress reports every year. There's a due date for them. We read them. We have to determine if the progress you've made during that year is satisfactory to get the next year's tranche of money. And sometimes you will have unobligated funds in excess of 25% of the award, in which case you have to explain to us what you plan to do to reduce that to manageable amounts. We also give you advice and permission on no-cost extensions, which means you still have money in the grant after the period of award is ended, but you're continuing the research and spending down the money. The first year is automatic. The second year requires more consideration, and the third year no-cost extension is highly unlikely. And we also give you advice on supplements. If you're submitting an administrative supplement, which is reviewed by program officers, we, the program officer, have to accept it for consideration, and we don't necessarily do that. That's not automatic. Again, speak to program officers for the details on that, and then we guide you to funding opportunity announcements as well, where they are appropriate. The scientific review officer governs the review process. They are highly knowledgeable. They have a number of rules that they are required to conform to. The reviewers are required to conform to those, and you as the applicant are also required to conform to them. One of the biggest rules is confidentiality. Do not try to contact anyone you think is on a review panel before or after the review. That will cause not just problems for you if it becomes known, but for the reviewer as well. It's a matter of misconduct. And the review officers will also ask you to serve on review panels. It is extremely valuable to do so. It's not simply as public service, but it also gives you direct insight into the review process, how people think, how people react to each other during review, and what sorts of things reviewers tend to pick up on as strengths and what sorts of things appear to be weaknesses. That will help you in the grant writing. There are programs as junior faculty that will help you enter the process of being a reviewer. And it's a good idea. And then the grants management specialist, every institute at NIH has them, and your grants management office at your institution are the people who interface with our grants management specialists. and they deal with the arcana of details of the awards and spending rates and things like that. But your program officer is always ready to help you if you have any questions anywhere along the way. So I'd like to turn now briefly to the Division of Aging Biology and to give you some overview of what it is that we do. We support basic, applied, and translational research. Basic research, you all know, is on mechanisms by which biological processes take place. Applied research could be thought of as the technology that goes into the study of biological processes, but it also supports biotechnology companies that are seeking to develop therapeutics or analytics or assays for biological change. And then translational research, which is taking research from laboratory animals or wild animals or domestic pets and trying to bring that to the clinic for the benefit of human health, because we are the National Institutes of Health and our primary concern is human health and well-being across all facets of what it is to be a human being. So we support research on the molecular drivers of aging in the division. These are often thought of as the hallmarks of aging, but it's also specific molecular pathways that often comprise genetic components of aging. And in those cases where aging features are heritable, as in centenarians, we're interested in understanding how those outliers, those especially successful agers, manage to do that. But also normal aging and also what leads to pathologies during aging. So we also are looking to manipulate the rate of aging. Aging had been thought of as something that's unidirectional. By and large it is, but the rate at which that direction, we go in those directions, can be changed. And we know this from quite a few studies. Among the most interesting, I think, are those of heterochronic blood exchange, taking blood from a young mouse or organism and putting it into an old organism of the same genus species and strain usually. And you find that some aspects of aging are slowed down, or taking blood from, or plasma or cells from an old mouse and putting them into a young mouse. Typically it's done that way. And you find that there's accelerated aging, that is aging phenotypes you'd expect in a late life mouse organism occur in the young. And we support translational research to support health at older ages. We have 4 components to the Division of Aging Biology. We are really organized around the convergence on the rate of aging. The genetics and cell biology branch looks at interactions among the hallmarks of aging. Applied physiology branch looks at mechanisms of physiological decline with age. It's geared more towards specific tissues and a number of pathological states. In the biological resources branch, it's the focus for research is on the development of aging lab animals for research. But the biological resources branch also has contracts to support biological resources for research, the most famous of which, of course, is the aging rodent colony. And the Office of the Division Director emphasizes rates of aging in human populations. We actually have a fairly good portfolio of grants where humans participate in the research as subjects of research. We are not exactly known for that, but we do have robust presence there. And also in the area of geroscience, which is taking basic biology where it intersects with the biology of dysfunction and the biology of disease. And that's part of the, really it's geared towards translational research. And shown in the middle are some of the technologies and findings that are involved. There are cell painting assays, which Dr. Carpenter, I believe at MIT, has developed. And shown here, it's a way of looking at multiple cellular phenotypes through microscopy. And that's something that we feel should be applied in the biology of aging. In the lower right corner are some results from the Wiscore lab looking at waves of changes in the proteome across age. This is a cross-sectional study, but it tells you a very important outcome. The changes during age are not obviously linear. Proteins may come and go. There's a sexual dimorphism in the amplitudes of these changes and in the timing of these changes. So it's pretty important, I think, to understand that some aspects of aging are not just comparisons between the young and the old, but they need to be looked at across the duration of life. And we also look at associations between, or our investigators look at associations between components of any analytical method. And from these types of plots, you can actually construct networks which allow you to discover various sorts of interactions and their strengths. And of course, there's the famous T-SNE plot, which allows you to look at heterogeneity in cell types. And I believe this specific plot comes from the tabula muris, which was a Stanford group that looked at cell heterogeneity in many tissues in mice. And they also have the tabula muris senis, which is the it's a program to look at cell heterogeneity with aging, and a very important prospect. In 2013, Lopez Otin and colleagues put forward a document on the hallmarks of aging based on hallmarks of cancer as a concept. It's a way of organizing, thinking about biology of aging, and it's very useful. It covers levels from molecules to organelles to cells to organs. And the key features of hallmarks of aging are that they change with age, that if you exacerbate a hallmark of aging, you increase the rate of aging, and if you do something to ameliorate or relieve the impact of an aging hallmark, you can slow the rate of aging, which means that you might delay the onset of a disease or dysfunction. We might reduce its severity, which is in essence the neuroscience hypothesis. We also support some significant programs that are foundational for research. The intervention testing program using mice. They're relatively heterogeneous mice from 3 inbred strains. And these are to test interventions for lifespan and also to archive samples, provide the data on the lifespans, and also to do some analysis of health, because it's generally been observed that where interventions extend lifespan, the older mice tend to look healthier and function better. And there's the Sener-Habditis intervention testing program, whose mission is to test in reproducible fashion the impact of interventions in genetically diverse setting. So 3 strains of each of 3 species of Sener-Habditis are used. They are quite diverse genetically. They represent more genetic diversity than is present in the human population at this time. So that's a test for robustness of an intervention. They both provide what could be called ground truth, with the idea that the field would then pick up that information for mechanistic studies. And then we have the Nathan Shock Centers, which are for excellence in the biology of aging. They provide pilot project funds, and they provide technical expertise, and they provide resources for data analysis. And we now have 8 centers. And you can reach that, find out about those centers just by looking for Nathan Shock Centers on your web browser. So I'm the director of the division. Dr. Stacy Carrington-Lawrence joined us just this past May. Her mandate is to promote research in human subjects, especially in the area of health disparities, and also to help us develop more outreach on training programs for the next generation of science. The aging physiology branch currently has 3 members. We will be expanding this year to 5. We are actively recruiting. Rebecca Fuldner is the branch chief. John Williams and Candace Kerr are 2 excellent program officers. Rebecca's area of expertise is immunity and inflammation, and also interest in microbiome, and has part of the portfolio on HIV that is held at NIA across all divisions. John Williams, all things bone and muscle are of interest to John, and as well as attempting to promote research on kidney function and failure with age. Candace works in the cardiac and vascular structure and function portfolio, endocrine, exocrine, and reproductive systems, as well as respiratory systems and in stem cell aging and aging of the niche. In the biological resources branch, Francesca Macchiarini is the chief. Manuel Moro is the program officer with a degree in veterinary medicine, as well as master public health and a doctorate degree in research. And Heidi Brogdon runs the rodent ordering system. So if you want to order rodents, you'll meet Heidi. And Francesca and Manuel manage the biological resources contracts. And Francesca is the science officer on the intervention testing program, and her portfolio is in resilience, developing analytical tools for measuring resilience in laboratory animals. Manuel's passion is in non-human primates, and his portfolios are in the development of new models for aging research and in comparative biology of aging. He has helped to get marmoset husbandry better handled at the various rodent centers and other laboratories around the country, and also has developed a program on looking for novel non-rodent models for Alzheimer's disease, which is a major area of interest in the NIA. And this is the genetics and cell biology branch, and Max Guo is the branch chief. Yue Friedel and Viviana Perez are the very good program officers who came to us from academic careers more fairly recently. And I won't read everything that's on here, but Max's expertise is in genetics, epigenetics, and all things nucleic acids. And Yue Friedel's work is in metabolic regulation program, supporting investigators on mitochondrial fitness and integrity. And Viviana Perez came to us most recently from the University of Oregon, where she was working on proteostasis, cell senescence, autophagy, and had quite a successful career. We're quite delighted that she came to us to work as a program officer. So that's our group, and thank you very much for taking the time to listen to this presentation. I'll be available at the round table later on and during the meeting. And I'm always available if you want to email me with any questions about the division. Where it's possible, I will either answer them or I will direct you to the appropriate person. Thank you very much for your attention.